Today in this lecture, we will be talking about the basic structure and organization of chromosomes. Now, as we all know that cells have the capability to give rise to new cells. And by giving rise to new cells, what cells do is that whatever information is encoded in their genetic material, in their hereditary material, that information is passed on from one generation to another. So what is this information? This information will be regarding the structure, function, overall development and differentiation of a living organism. And we all know this information is encoded in the genetic or hereditary material of the cell. Now, how are the genetic materials in prokaryotes as well as eukaryotes? Where are they located? How are they concentrated in the cell? So, prokaryotic genetic material is usually concentrated in a specific clear area present in the cytoplasm. That area is known as a nucleoid. The bacterial chromosome is a single circular double-stranded DNA molecule which is attached to the inner side of the cell envelope at a specific point. For the packaging of the prokaryotic genetic material, there are no histone proteins. Instead, they have specific proteins for packaging, and those proteins are known as the HU proteins. The Escherichia coli DNA is a circular molecule that has a size of 4.6 million base pairs, and it contains 4,288 protein coding genes whose location is known. So there are 4,288 annotated protein coding genes which are organized into 2,584 operons. There are 7 ribosomal RNA operons as well as 86 transfer RNA genes in the prokaryotic genetic material. Now there are certain exceptions like bacteria, Borrelia burgdorferi. There are certain Streptomyces species which possess linear chromosomes like the ones we see in eukaryotic system. Now, apart from the chromosomal DNA, bacteria also carry certain extra chromosomal small circular closed DNA molecules. These DNA molecules remain floated in the cytoplasm and are known as plasmids. Now these plasmids, they are not indispensable to the host, but they will confer certain unique characteristics to the host in which they are present. And these characteristics are due to the different genes that are present on different plasmids. So according to what genes are present on the plasmids, we classify these plasmids into F plasmids, R plasmids, there are col plasmids, virulent plasmids, and degradative or metabolic plasmids. So this is how the prokaryotic genetic material is arranged. As you can see in this image, at a particular point in the cell membrane, they are attached. It's a folded structure. The folding is due to binding of specific proteins which are known as HU proteins. And this DNA therefore is present in the cytoplasm in a specified clear area known as a nucleoid packaged with proteins. In this second image, we can see these floating small circular molecules. These DNA molecules are plasmids. Now, as against the prokaryotic genetic material, eukaryotic genetic material is linear double-stranded DNA molecules, and are present enclosed within the nucleus. They are present in the compartment, and that compartment is the nucleus. Now, on these chromosomes, genes or hereditary units are located. Now, these chromosomes, they vary in shapes during the different phases of the cycle of the cell. They can be thread-like in appearance. At the time of cell division, it becomes condensed, it becomes highly packaged into structures that are known as chromosomes. So what are chromosomes? Chromosomes are rod-shaped, filamentous bodies that are present in the nucleus and become visible only during cell division. They are the carriers of genes that are the units of heredity. Chromosomes are not visible in active nucleus due to the high water content. But at the time of cell division, when they are in the most packaged, most condensed form, they are clearly visible. Walter Fleming, who is a German biologist, as well as the founder of cytogenetics, around 1880s, revealed that during cell division, the nuclear material organizes itself into visible thread-like structures, and these thread-like structures can be stained with basic dyes. The term chromosome was coined by Walder in 1888, and Walder was a German anatomist. Why he named them chromosome was because these bodies could be stained, they could be colored. So chromosome comes from two terms, chrome, which means colored, and soma, that means body. So literally it means colored bodies. Chromosomes can also be defined as higher order organized arrangement of DNA and proteins which contain many genes or hereditary units. Along with these genes, it also contains the regulatory elements as well as other nucleotide sequences. Chromosomes contain DNA bound with proteins. These proteins in eukaryotic system are the basic proteins known as histones, which help in the packaging of DNA, to condense the DNA, to fit it into the nucleus, and also to control the functions of the genetic material, that is replication as well as gene expression. Chromosomes vary both in number as well as structure among organisms, but for all the individuals of a particular species, the number of chromosomes will remain constant. So it's a characteristic value for every species. This was reported by Bendon and Bowery in 1887, that number of chromosomes in each species is constant. In 1902, Sutton and Bowery suggested that chromosomes are the physical structures which act as messengers of heredity. Now in eukaryotes, to fit the entire length of DNA in the nucleus. Now if you open up a DNA molecule, it has a length of approximately 2 meters. So such a long DNA molecule has to be fit into a nucleus, the diameter of which is just few micrometers. So to fit this entire DNA in the nucleus, it undergoes condensation. The degree to which a DNA condenses is expressed as its packing ratio. So what is packing ratio? It is a length of the DNA divided by the length into which it is packaged into chromatin along with the proteins. So DNA is packaged stepwise into higher order chromatin structure This packaging structure is known as hierarchies of chromosomal organization. There are normally 2 copies of each chromosome present in every somatic cell. The number of unique chromosomes that is represented by N is known as its haploid number. Now the haploid set of chromosomes is also known as the genome. The total number of chromosomes represented by 2N is the diploid number. Now ploidy, in haploid as well as diploid, this term ploid, it refers to the sets of the chromosomes. So there is one set of chromosome in haploid and there are 2 sets of chromosomes in diploid. Apart from the nuclear chromosomes, in eukaryotes, chromosomes are also present in mitochondria as well as in chloroplast, which are semi-autonomous organelles. They have their own genetic material. The organism which till date has been reported to have the lowest number of chromosome is the nematode, that is Ascaris megalocephala univalens. It has only 2 chromosomes in the somatic cell, that is 2n is equals to 2. In diploid cells, there are 2 of each kind of chromosomes. They are known as homologous chromosomes except for the sex chromosomes. So for example, in humans, there are 23 pairs of homologous chromosomes, or the 2n or diploid number will be 46. In human females, there are 44 autosomes, that are the somatic or the body chromosomes, and 1 pair of homomorphic sex chromosomes, that are X and X. Therefore, homo means same, morph means form. In contrast, human males have 1 X and 1 Y chromosome as sex chromosomes, and therefore human males are heteromorphic sex chromosomes, and they have 44 autosomes. That is common. The size of a chromosome is normally measured at the time of metaphase, because that is a stage where the chromosomes are most condensed, and because they are most condensed, they are easily visualized at the time of metaphase. Now, size of chromosome ranges. It can be as short as 0.25 micrometers in fungi and birds to as long as 30 micrometers in some plants such as trillium. The mitotic chromosomes fall in the range of 3 micrometers in Drosophila melanogaster, that is fruit fly, to 5 micrometers in man and 8 to 12 micrometers in maize or corn. In general, monocots contain large sized chromosomes as compared to dicots, and also organisms with less number of chromosomes contain comparatively large sized chromosomes. As I said, shape of a chromosome will change from phase to phase. So when it is in the interphase stage, that is when it is not dividing or it is in the resting stage of the cell cycle, chromosomes, they are present in the form of thin, coiled, elastic, contractile, thread-like structures which are known as chromatin. These thread-like structures, they condense further at the time of cell division. Therefore, at the metaphase and anaphase, the chromosomes, they are thick and filamentous. Now each chromosome contains a clear zone known as centromere with a kinetochore along their length. This centromere, it divides the chromosome into two parts. These two parts are known as chromosome arms. The short arm is designated as the P arm, where P coming from petite, and petite means small. The large arm is known as the Q arm. So where this centromere is positioned on the chromosome gives the chromosome its different shapes. So chromosomes based on the position of the centromere could be telocentric, where the centromere is present on the proximal end of the chromosome. Such chromosomes are also known as eye-shaped chromosomes. Acrocentric, where the centromere is present at one end, giving it a very short arm. P arm is very short and the Q arm is very long. Such chromosomes also appear like a J, so they are known as J-shaped chromosomes. Submetacentric chromosomes, where the centromere is present near the center. These are also known as L-shaped chromosomes. And metacentric chromosomes, where the centromere is placed at the center. These are also known as V-shaped chromosomes. A chromosome at the time of metaphase, it contains two symmetrical structures. Now remember, the chromosome, the DNA is already replicated during the S phase of interphase. So it's already present here as two symmetrical structures. Each structure is known as a chromatid. So there are two sister chromatids which are joined to each other only at the centromere, and they become separated at the beginning of anaphase. Now each chromatid is one DNA molecule. The chromomeres are bead-like accumulations of the chromatin material that are sometimes visible during the interphase chromosomes. These chromomeres bearing chromatin gives the appearance of a necklace in which several beads occur on a string. These chromomeres are especially prominent in giant chromosomes like the polytene chromosomes, and they are regions of tightly folded DNA. Centromere in the chromosome contains specific DNA sequences to which special proteins attach, giving it a disc or a cup-shaped appearance. The structure is known as a kinetochore. When observed under the electron microscope, this kinetochore appears as a plate or cup-like disc that is 0.2 to 0.25 nanometers in diameter. The kinetochore is a position in the centromere, the specific sequence with proteins present in the centromere to which the spindle fibers attach at the time of cell division. Chromosomes of most organisms, they contain only one centromere. So a chromosome having one centromere is known as monocentric chromosomes. Some species, they have diffused centromeres. That is, the microtubules will attach along the length of the chromosomes, not at a particular point. Such are known as holocentric chromosomes. So they have diffused centromere. One of the examples of chromosome having diffused centromeres is Ascaris megalocephala, that's a nematode. It's an example of diffused centromeric chromosomes. Telomere is another important portion of the chromosome. These are the ends of the chromosomes which help in the completion of replication, as well as they provide stability to the chromosomes. They prevent other chromosomal segments to fuse with them. Besides the primary constriction is a centromere. So besides the centromere or primary constriction, chromosomes also possess secondary constriction at any point of the chromosome. These secondary constrictions, they are constant in their position and extent, and in which chromosome they will be present in a particular species. Therefore, these constrictions help in identifying particular chromosomes in the set. Chromosomes also contain nucleolar organizer regions, which are secondary constrictions which contain genes coding for the ribosomal RNA. That is the 5.8S rRNA, 18S rRNA and 28S rRNA. The ribosomal rRNA or the ribosomal RNA is formed in the nucleoli. So chromosomes contain nucleolar organizing region which induce the formation of nucleoli and are associated with the biosynthesis of the ribosomal RNA. Sometimes the chromosomes bear round, elongated or knob-like appendages. These appendages are known as satellites. The satellite remains connected with the rest of the chromosome by a thin chromatin filament. Chromosomes having satellites are known as sat chromosomes. Now, based on the position of the centromere, chromosomes are categorized into 4 types. Metacentric. In this type of chromosome, the centromere occurs in the center. Chromatids are of equal length. So there is no differentiation. There is no P and Q arm. Both the arms will be equal. Submetacentric. In this type of chromosome, the centromere is little away from the center. Therefore, chromatids of one side are slightly longer than that of the other side. So there is a differentiation in the P and Q arm. Acrocentric. In this type of chromosome, centromere is located close to one end of the chromatid. Therefore, the chromatids on opposite sides are very long. A small round structure attached by a very thin thread is observed on side of the shorter chromatid. This small round structure is termed as the satellite. The thin strand at the satellite region are termed as NOR or the nucleolar organizer region. In humans, acrocentric chromosomes are chromosome number 13, 14, 15, 21 and 22. Finally, we have the telocentric chromosomes. In this type of chromosome, the centromere is placed at one end of the chromatid and therefore they have only one arm. Telocentric chromosomes are not seen in humans. They are seen in the house mouse chromosomes. Now this image shows the 4 different types of chromosomes based on the position of the centromere. So they are metacentric, submetacentric, acrocentric and telocentric. The I-shaped chromosomes, J-shaped chromosomes, L-shaped chromosomes and V-shaped chromosomes. Karyotype. Eukaryotic species have got several chromosomes that are detected only during division, that is during mitosis or during meiosis, and they are best observed during the metaphase stage because at this stage they are in the most condensed form. Now each eukaryotic species is characterized by a karyotype. Now a karyotype is a numerical description of the chromosomes in a normal diploid cell. In a karyotype, we arrange the chromosomes on the basis of their size. That is, the biggest chromosome first and the smallest chromosome last. So on the basis of their number and on the basis of their size, chromosomes are arranged, and such diagrammatic representation on the basis of their number and size is known as a karyotype. A karyotype is important because genetic research can correlate changes in the karyotype with changes in the phenotype of the individual. So certain disorders can be studied with the help of the karyotype. Now in this image, the left side, it shows a normal human karyotype. On the right side, what we see here is chromosome number 21 is present in 3 copies. So this condition is known as trisomy 21, also known as the Down syndrome. So a karyotype helps us correlate changes in the karyotype with changes in the phenotype of the individual. Now a karyotype of a species, when represented diagrammatically such that it shows all the morphological features of the chromosome, is known as a ideogram. So this left image is a karyotype, and the right image, which shows all the morphological features, the bandings of the chromosomes, is known as the ideogram. So that is all about the basic structure and organization of the chromosomes. Thank you.